Okay, let's start our Carbs Concept Map. So if we scroll in on the top left, we're looking at our carbohydrates. Carbohydrates are found in ring structures that are called hemiacetals. That's if they contain aldehydes. Or if they contain ketones, they're called hemiketals. We can separate our carbohydrates based off of their chain length, like we did with our proteins and our lipids. If we have one sugar, it's called a monosaccharide. The most common ones that we talked about were glucose, galactose, and our fructose. And just to point out that our galactose and glucose are epimers of each other. Our 2 sugars long are called disaccharides. And both the monosaccharides and disaccharides both taste sweet to us. With our disaccharides, we have several different ones that we had discussed. We had maltose. Maltose is glucose, alpha 1,4 glucose. Isomaltose, which is an isomer of maltose. Both are breakdown products of starch and glycogen digestion. And isomaltose is glucose with the alpha 1,6 branch point stuck in between. So it's glucose alpha 1,6 glucose. Lactose is your galactose, and it's a beta 1,4 glucose. And then sucrose is glucose alpha 1 beta 2 fructose. Our disaccharides and all of our carbohydrates are joined together by glycosidic bonds. The glycosidic bonds that are located in between these carbohydrates are made by enzymes that are called glycosyl transferases. Our next chain length are the oligosaccharides, which are 3 to 10 sugars long. If you remember, we can't digest or absorb them, so they're more fermentable. And you'll find those in your beans. Anything longer than 10 sugars is called a polysaccharide. And we had several different polysaccharides that we had talked about. With our the storage form in plants is known as your starch. So these are more storage functionality of them. Starch, which contains a mixture of amylose and amylopectin. Of course, starch is a homoglycan of glucose, so it's all glucose. Amylose is glucose with only alpha 1,4 bonds in between. And amylopectin is alpha 1,4 bonds, and then we have branch points of alpha 1,6 that are found every 24 to 30 glucoses. Glycogen is very similar to the amylopectin structure. It's a storage form of carbohydrates in mammals. And it has, again, mostly alpha 1,4 bonds, when you have alpha 1,6 branch points that are located every 8 to 12 glucoses. So much more heavily branched structure compared to your amylopectin. With glycogen, we can store it in the liver and we can store it in the muscle, but stored in the liver, it's more important for blood glucose regulation. If we push it in a muscle, the muscle has to use it, so it's going to be used during exercise. Other polysaccharides, we had dextrans, which are made by bacteria. It's usually found in the mouth. And those dextrans are highly concentrated in dental plaque. They make it really sticky, very adhesive. And then chitin was a structural component of the exoskeleton of arthropods. So there wasn't anything super exciting to note about chitin other than it is highly sulfated. When it comes to the structure or the functions of these compounds, we have the storage, we have energy, and then we have signaling. So some of the other functions include structural components, especially the extracellular matrix. With the structural components of the extracellular matrix, we have our glycosaminoglycans, or GAGs. And GAGs, we have a couple different ones to note. we have our hyaluronic acid, which contains glucuronic acid and N-acetylglucosamine. That's why glucosamine supplementation is beneficial for synovial fluid patients with osteoarthritis because it's a precursor for hyaluronic acid. And of note with hyaluronic acid, it's the only GAG that is not sulfated. So a sulfate contains a negative component, and your sugar acids are also contain a carboxylic acid, which would have a negative charge associated with it at physiologic pH. The most abundant type of GAG in the body is going to be your chondroitin sulfate. Other ones, we have keratin sulfate and dermatin sulfate. So your keratin sulfate's found in your hair and your nails, and dermatin sulfate's in the skin. Of course, your glycosaminoglycans are just repeating disaccharides. It's that sugar acid and sugar amine that are going to be repeated over and over again to make your GAGs. If GAGs are attached to proteins, they're called proteoglycans. And the proteoglycans are found in the extracellular matrix. They're found in discs, and they're found in synovial fluid, among other places. If you recall with our proteoglycans, they can either be a single bottle brush structure, or, so that would be a monomer, or we can have hyaluronic acid with a bunch of different bottle brushes attached to it to make an aggregate. Other components of the storage components include storage forms or structural components of plants. And the structural components of plants are fiber. It's going to be on the top right hand over here. So with fiber, on the nutrition label it's listed out as dietary fiber, which is inclusive of all types of fiber. We can separate them into several different groups. We have insoluble, which really don't do much. They don't swell, so it does not swell in water. But they do increase gastrointestinal motility, so it's going to speed up movement. Good for your constipated patients. So the most common one that we talked about was cellulose, which is most of the fiber that we do eat. And you'll find it in your peels of your fruits and vegetables. You'll find it in bran as well. Your soluble fibers are those that do swell in water. And of the soluble fibers, some are viscous, some are fermentable, some can fall into all categories. Some that are viscous are more likely to have an increase in satiety, which means makes us feel a little bit more full. And remember, there's only about 22% of all fibers do have that effect, that satiety effect. Some examples of our viscous fibers would include things like your pectin. And pectin is a heteroglycan. I'm just going to put HE here. Heteroglycan, as it's made of multiple different sugars. It's a galacturonic acid and then a weird L-rhamnose sugar that's every 25 sugars. It's found on the insides of your fruits. We have another heteroglycan that's actually a lot of different compounds that are kind of lumped together and collectively referred to as gums. These are things that you'll get from trees, from seeds, bacteria, naturally found in your oats and barley. Oats and barley also contain your beta-glucans, which are really viscous. This is a homoglycan of glucose, but it's beta-1,2 and beta-1,3 bonds that we can't digest. So some of these soluble fibers are also fermentable fibers. If we can ferment them, we can turn them into gas and short-chain fatty acids. If we can ferment them into short-chain fatty acids, those fatty acids can be absorbed within the colon, and we can get about 1.5 to 2 calories per gram compared to the 4 grams that we would normally get from starch and glycogen, or 0 grams or 0 calories per gram that we would get from cellulose. So this is your form of colonic salvage, that we're able to salvage a little bit of energy in the colon through fermentation. When we ferment, it's also a food for that good bacteria, so it's known as a prebiotic. Probiotic is the actual bacteria, and then prebiotic is its food. Some examples where I have lines drawn here: pectin, gum, some of the beta-glucans are also fermentable. Another one, which is referred to as fructans. Fructans are inulin and fructooligosaccharides. Inulin's a little bit bigger. Those are the ones that are 11 to 60 fructoses. And fructooligosaccharides are 2 to 10 fructoses. So your fructans are homoglycans, but they're homoglycans of fructose, which is different. All of the other homoglycans we've talked about are homoglycans of glucose. I'm going to scroll back up here. I forgot to write, for cellulose, it's a homoglycan of glucose as well. So the last part of this is the last function, which is signaling. Our glycoproteins are types of signals, carb types of signals in the body. This is where we have an oligosaccharide attached to a protein. And the oligosaccharide is what's going to give its specificity to that signal. It's going to be based off of the types of sugars, as well as order of attachment, what bonds are located in between, as well as how many bonds are located there. So that's your cell surface recognition. This is also what gives us our blood types, whether you're A, B, or O. And then these are also referred to as, some are mucins, which are lubricants and mucus as well. Other signaling compounds are lipopolysaccharides, which we said were inflammatory signals, and these are made by gram-negative bacteria. So super inflammatory, those are ones that we absorb along with all the rest of our lipids, since they are mostly lipids, and we'll transport them as part of our chylomicrons as well. And last but not least, we have receptors. The receptors for some of these, the oligosaccharides from the glycoproteins, are called lectins. And these lectins will help mediate those cell-cell interactions. There was a type of subcategory of lectins that we had talked about that were called selectins. So these are protein receptors that will attach to the oligosaccharides that are acting as signaling compounds. So that's how we read the sugar code, are through these selectins and lectins. Thank you for listening.